Hello, my name is Keren. I was born here in Israel, in a small kibbutz in the south of Israel, not far away from Gaza. I came to Jerusalem since I wanted to study my first degree at the Hebrew University. I stayed for my second degree here, luckily for me, since I met my husband during that time. I'm studying here at IMRIC, which is a great place to study, and since I like it, I stayed it for my third degree, which I'm now finishing. My research is about genetic in bacteria, and the good thing about it is that bacteria in a lot of ways is a good model organism which is very close to human, and there's a lot of systems that actually work the same, and it's just easier to study it in bacteria than in human. On the other hand, systems that are different from human could be the new target for drugs. I try to combine spending time with my family, since I also have a son. He's 2 years old by now, and try also to enjoy my research and spend as much time as I can with that. In February 2011, we published in Science an article about messenger RNA localization in bacteria. The breakthrough with that is that if we look at the systems, and DNA is actually being transcribed, or we will say translate, to RNA, and then RNA is being translated to a protein. So in order to get a protein from a gene, we need the messenger RNA. And the new breakthrough about it is that in the last 20 years, it is well known that there is localization of messenger RNA in human cells and in other high organisms. It wasn't known that there is a messenger RNA localization in bacteria cells. This is the breakthrough. The bacteria cells are simpler, easier, and messenger RNA localization is a very, very complicated thing. So in order to well understand it, it's easier to study it in a model organism which is simple, multiply very, very fast every 20 minutes, easy to do all kinds of genetic modifications and changes. So it's actually easier to study this system in bacteria than in human. If we will understand it in bacteria, we will be able to understand it in human. If there's change between the bacteria system and the human system, it's also good. Because if the bacteria system is a bit different than the human system, we can target new drugs to the bacteria, and since antibiotics soon is not going to work, maybe we will find new antibiotic that can work, or design new antibiotic to the differences between those species. My goal was actually to understand a system in bacteria, and we found out this new breaking thing which we didn't think that's what we would find, and we actually found out that bacteria is a suitable model in order to study messenger RNA localization. So that's actually a new thing. I'm studying it for the last, well, eight years since my master's degree. If you find something new in science, the first thing that you do, you recheck yourself like 10,000 times to see that you don't get some artifact. That's why we did the same thing in different systems. We used 3 different systems. We took biological approach. We look at live cells in the microscope, and that's why we did it in a few ways. And yes, once you find out that you have something new, something exciting, something that nobody else found before, then of course you're happy. I would like, of course, to continue working on this system, at least until I will finish my PhD. Then I would really be happy if I would be able to take my family, which is not easy, abroad for a post. I am looking forward to be a professor, and of course I want to go back to Israel and be a professor here, if not at the Hebrew University, at least in another university in Israel. I really want to continue working on bacteria, and I just think they are great model in order to study all kinds of things, all kinds of systems, and I would prefer continue working on them. And yes, I see myself, if not winning the new Nobel Prize, at least then I will be able to cure a new disease or find a new antibiotic.